Okay. How many parts to the immune system? 2 parts. What are they? Specific and nonspecific, and we always start with nonspecific immunity. Nonspecific immunity is the kind of immunity that basically tries to fight against anything. That's why it's called nonspecific. It does not need to know what it's fighting. It could be a splinter. Does your body try to keep a splinter out? Yes. Does it try to keep a sneeze out? What do you think? Somebody else's sneeze. Does it try to keep somebody else's sneeze out of you? Yes. Does it try to keep little rabid raccoons out of you? Yes. So does your body care if it's a raccoon or a sneeze or a splinter or a staple? No, it just wants to keep everything out. And the part that wants to keep everything out is nonspecific immunity or innate immunity. We will talk about the type that has to identify the specific pathogen in a minute. Okay, as we said, which I'm repeating to make sure it's all in the lecture, that nonspecific immunity, the first part of that, is known as the first line of defense. It consists of the skin and the mucous membranes, as well as any associated chemicals or secretions. Everybody knows what the skin is already. We don't really need to say much about what it is because you already understand what it is. How is it a barrier, though? It's a physical barrier. Absolutely, it's a physical barrier. Made up of what stuff? How is the skin a barrier? Just the skin part. What'd you say, Andy? Cells. What do you mean by cells? You remember the 2 parts of your skin? Dermis and epidermis. What's the epidermis made of? Cells, right? Epithelial cells. Multi-layered epithelial cells that flatten out on the top because they're full of keratin and they have died. So the epithelial part's called stratified squamous epithelium. You remember that? It's kind of thick. Underneath that, we have the other part of the skin called the dermis, which is connective tissue, which is even thicker, much thicker than the epidermis. So you put all that together and you get a skin which is kind of thick and provides a nice little barrier to the outside world for us. Additionally, we can say that on the skin, aren't there secretions that help us to like fight bacteria and fight fungus? Do you know what kind of secretions on the skin help? Pardon? Did you say wet?
No.
What? Sweat. Oh, yeah, actually sweat does have some chemicals that are antibacterial in it. And the pH of your skin, this is important, the pH is acidic, which, as we know, certain bacteria will die off in an acidic environment. There are also chemicals called lysozymes. Lysozymes. And what lysozymes do is, well, they will break stuff down. Because what does the word zyme tell you? It's an enzyme. Do you remember what lysosomes were? By chance from AMP1? They were little vesicles inside the cell. So what lysozymes are is chemicals that are secreted in body fluids, like in saliva and tears and other body fluids as well. And they contain enzymes that destroy bacteria. The oil in your skin actually contains antibacterial and antifungal properties. And then we have mucus, which would lead me to talk about mucous membranes. Where are your mucous membranes? Okay, your oral cavity, right? Leading into the respiratory area, so the nasal cavity and back, your throat, the inside of your digestive tract, all the way down to the anus, the inside of the reproductive system as well. All of those are considered mucous membranes. One of the main jobs of mucus is to do what? Simply to trap stuff. Mucus is thick and sticky. Its job is to trap. So if we're talking about in the respiratory tract, which you already had a little bit of lab on this, tell me what kind of epithelium lines the trachea? Pseudostratified ciliated columnar epithelium. It's got the cilia. What do? Which way? They push up. And what they're doing is they're pushing the mucus and whatever happens to be trapped in the mucus. They're pushing it up so that it can go where? Back and down your esophagus into your stomach, where the stomach has a really acidic pH and digestive enzymes to break down that harmful stuff. So look, that, even the stomach, is a mucous membrane in there, right? Inside the internal aspect of the stomach. So it's kind of all working together. Do you think that the stomach goes, I wonder what kind of bacteria you are. I wonder if you're a splinter. I wonder, its acid is just there to destroy whatever is there and to activate chemicals, which we'll talk about when we get to digestive, which we haven't done yet. So the big concept is these physical barriers and these chemicals, they don't really care what they're attacking. They just try to destroy stuff that's living that could harm us or try to keep stuff out of the body. That's the big picture on that. Then we go a little bit further. And we say that in addition to the skin and the mucous membranes, if we go inside the body, there are certain kinds of cells that like to fight for us. What kind of cells would you associate with cells, or which kind of cells do the fighting inside your body? White blood cells. White blood cells do. So we will be talking about a few different types of white blood cells. Okay? Additionally, and you see it kind of generic up there on this slide. Additionally, we will be talking about some chemicals that do not necessarily need to identify the agent to destroy it either, and those are the chemicals you see here. So what we're going to do now is we're going to go through each one of these that you see on this slide. We'll begin by talking about the phagocytes. What is a phagocyte, first off? And what does phagocyte mean? Okay, it's a cell that eats, right? It's a cell that eats. It's probably the name of the phagocyte that we are most common with would be the macrophage, which is a white blood cell, as we know from lab, was a monocyte, got activated and turned into a macrophage and roams through the tissue to gobble up foreign debris and microorganisms and all kinds of stuff. Just to gobble up and destroy the harmful material, the macrophage does not necessarily need to know exactly what it is. So in this case, as we talk about it as a phagocyte like this, it is part of non-specific immunity because it doesn't need to identify if it's a bacteria, if it's a virus, if it happens to be a parasite, whatever it is, it will just take care of it. Now you can see from the slide that there are macrophages that live in different places of the body. Some of them spend their entire life in a certain organ, so they kind of call them fixed macrophages. And I want you to know these two examples right here. In AMP1, you should have mentioned microglia. Anybody, does that ring a bell for anybody at all? It was one of the neuroglia in the central nervous system. So what microglia are is they are actually macrophages in the brain. White blood cells that live in the brain. And Kupffer cells are white blood cells that spend their lives roaming around in the liver. We also have some macrophages that instead of necessarily staying in one kind of specific location, they're free to wander around through the tissues of the body. And one of the examples that they use there would be macrophages that are found in the alveoli, which would be the lungs. Sometimes they refer to those as dust cells, D-U-S-T, dust cells. Lungs. Additionally, and I'll go ahead and mention this, neutrophils are phagocytic. They can work like phagocytes. See, macrophages aren't the only phagocytes. There's other phagocytes as well. Neutrophils, eosinophils are also phagocytic. I know your book doesn't happen to, I don't think it happens to mention those here, but they are. So I want you to understand that as well. So let's see what phagocytosis looks like. If you look right here, and let's pretend that this is a white blood cell. Just say it's a macrophage. What I'd like you to focus on is just this part at the top. So pretend, just for sake of argument, the rest of it's all nice and neat and smooth and you don't see anything else. So notice how it's forming almost what we would say looks like a little mouth, and it's kind of gobbling that up. What it is going to do is it actually attaches, and then you may remember this from another class or before in this class. It actually changes the shape of its membrane to reach all the way around and enclose the object. And then that would move into the cell. At this time, they say this, that this little vesicle that has those objects in it is called a phagosome. All right? Why? Because it's been eaten. It's been eaten and pulled in, so it's a phagosome. Now, the white blood cell's not going to just want to leave that intact. The whole point in pulling it in is so it can destroy it, right? So let's think back to AMP1. There's these little chemical sacs that are inside the cells that contain digestive enzymes. You see it on the picture. What's its name? It's a lysosome. So what happens inside a lysosome then is that, or inside the white blood cell, is that the lysosome and the phagosome fuse together. And those digestive enzymes can now destroy that bacteria or whatever it happens to be, those particular little agents there. And then in the end, it's kind of like people, right? Things come in one side, and what do? They move through our body, we digest them, break them down, and then they pass out at some point too. So you can see then eventually it has been broken down, and then it just gets released out of the white blood cell. And of course, the white blood cell doesn't just necessarily do this to one thing at a time. It can happen in lots of locations. It doesn't have one mouth and one digestive tract leading to one anus. It's a lot different than that. And you can see right here, this is an exceptional picture of what a macrophage would do. Notice how it reaches out its little processes. They kind of look like tentacles, And it can create new ones all the time, and they can get thicker or bigger depending upon what they grab a hold of. It's going to pull it back and just kind of envelop it and pull it in. All of a sudden it would be in a phagosome. It would fuse it over with a lysosome, break it down, destroy it, and spit it out the other side. Pretty cool. the next type of cell, which is actually different, is called a natural killer cell. Natural killers are not phagocytes, okay? They are not phagocytes. Natural killers are a type of lymphocyte. They are a type of lymphocyte. They're not one that we typically talk about, because usually when we talk about lymphocytes, do you remember the name of the two most common ones? I don't know if you did that yet. They're called T cells and B cells, right? The common lymphocytes are either T cells or B cells. Natural killers are different. Their job is to attack cancer cells and virally infected cells. Cancer cells and cells infected by a virus. They do so by releasing a chemical called perforin. 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 Anybody happen to know what perforin does? You won't see it up there. You probably see it in your book if you're looking at the old version of the book. The new book, they kind of took that word out. Exactly. Perforin is a chemical that pokes holes in a cell membrane. It perforates the cell membrane. If you put holes in a cell membrane, and we're talking about natural killer cells going out and poking holes in bacteria or trying to poke holes, in a cancer cell or a virally infected cell, what's going to happen to that cell? What's going to happen is fluid's going to rush into that cell. That cell's going to swell up and burst. Do you know what they call that when a cell swells up and bursts? It's called cell lysis. Cell lysis. So natural killer cells use perforin to cause cell lysis. Now, here's where I have to explain a little something. Notice that this is still non-specific immunity. How can we say a cell attacks virally infected cells, but it's not specific? Because it doesn't have to identify the particular virus. It could be any virus. It doesn't have to know what kind of cancer it is. Just cancer cells in general. So we can say that it targets cells that do not have markers of self on them. Okay, what do we mean by that? So let's set it up a little bit. What is a cancer cell? Okay, it's one of your body cells that has started to divide too rapidly. That's the basic idea. Cancer cells are associated with different proteins. Cancer cells have different markers on their surface than your normal cell, commonly. So they're not recognized as self cells. They are your own cells, but they're not recognized that way. So what a natural killer does is it basically attacks any cell that doesn't have a self marker. Commonly, cancer cells and cells that are infected by a virus, they do not have that normal self marker that your body cell should have. So that's what tells it to do the attacking. Okay, the other part? Natural killers enhance what is known as inflammation, which we're about to talk about. Natural killers enhance inflammation. So you can see this part. First, it was real easy. The innate or non-specific immunity is just your surface, your skin and your mucous membranes trying to keep stuff out. Whether that stuff has got into your mouth, let's say something's in your mouth and your body wants to keep it out, that means it doesn't want it to break the barrier of your cheek or of your throat. It doesn't want it to pass through that and get into the bloodstream or get in your tissues. As long as an object is in your mouth, in your esophagus, in your intestines, it is technically not in your body. That's a tube that passes through you, just as if my hand is forming a little tube here. Just like we would say the pen is not in my body. Technically, if I put it in my mouth, it's not in my body. It's in a little hole, a cavity, kind of like that, you know. Except this cavity is a lot longer and goes a lot further places, but there's an opening there as well, okay? So when we're talking about keeping it out, we're talking about not letting it pass through, not letting it break through the mucosa. Just as if we don't want it, bless you, just as if we don't want it to break through the skin. And that's why it was nonspecific. We don't want anything to break through. We don't care what it is, whether it's a pathogen, whether it's a splinter, whether it's a rabid raccoon. We don't really care. We just want to keep it out. So that was the first line of defense. Then we get to this part, which is called inflammation. And inflammation is known as your second line of defense. And the basic concept, before we talk about how inflammation actually works and what it is, the basic concept of inflammation is that it wants to prevent spreading. Inflammation is there to try to prevent spreading. And to set the stage for repair. Now, we usually say that there are 4 main signs of inflammation. In the way in the medical community they say the main signs is they say the 4 cardinal signs. Okay, the 4 cardinal signs of inflammation. You may have heard these before. Redness, swelling, heat, and pain. Redness, swelling, heat, and pain. Redness, swelling, heat, and pain. So inflammation is a chemically mediated process. There are many different chemicals in the body that can produce this inflammation, or different aspects of the inflammatory response. Some of the most important ones. Histamine. Histamine. Do you remember what kind of cell we've already said is associated with histamine? Because it's not the one that's listed up there. It's one of the white blood cells. Basophils. Good. Basophils. Right? There is a cell that's like a basophil that's out in the tissues. Its name is a mast cell. I want you to link in your mind basophils and mast cells. It's different, but it's like them. It does kind of the same stuff. So what both basophils and mast cells do is they release histamine. There's kind of 2 major things that histamine does. One of them is it increases a capillary's permeability. Increases capillary permeability. What does that mean? See, it's in this case, it means get out. It makes it easier for the fluid to rush out of the capillary and into the tissues. And if you want to get stuff out of the capillary and into the tissues, how are you going to get more fluid out? What are you going to do to the blood vessel? You're going to dilate it, vasodilation. So the other thing that histamine does is it stimulates vasodilation. Vasodilation. So picture this, if you will. Picture the surface of your skin and picture a blood vessel inside of that. Now a little bee comes up, flying around, and sticks his little tail in the surface of your skin and then takes off and dies, of course, right? And then there's little chemicals there. What happens to your skin? It swells and turns what color? And does it hurt? And if you touched it, would it feel warm? What is that process? That's inflammation right there. How does it work? Here's how it works. The body knows that that bee stinger contains some poison. And the body does not want it to spread. So what does it do? It releases instantly histamine that causes the blood flow to increase to that area and the capillaries to open up to push fluid into that area so it swells and it raises on the surface. All right? That's pretty cool. And it's not just histamine that plays a role in all of this. It's also there are different proteins in the blood. There are chemicals called kinins. Do you remember from before, whenever we see the term kinin or kine, what do we think of? There's just one generic thing I want you to think of when you think kinin. I want you to think of activation. Remember kinases? Protein kinases were chemicals that activated. So anytime you see a kinin, think about activation. They kind of activate inflammation. Prostaglandins and leukotrienes, any shock there that they would play a role in this? Notice I'm not getting picky. I'm not naming the names of prostaglandins and leukotrienes. I'm just trying to give you the idea of the kind of chemicals that play a role in this inflammation. And then a chemical called complement that we're going to talk about in just a minute. So complement will make a lot more sense in just a little bit when we talk about it. Therefore, if you think back to this, and you think back to the bee sting, and you think back to the tissue swelling around the bee sting, tell me what causes the redness of inflammation. The increased blood flow to the area. Blood's red, right? So the increased blood flow to the area causes the redness. That's cool. What causes the swelling? The increased capillary permeability, the fluid in the tissues, causes the swelling. What causes the heat? The increased blood flow. How hot's your blood? Do you remember from the blood lecture? 100.4. So you take a lot of blood to an area, it starts to heat up. So the only thing now left to understand is what causes the pain. Some of these chemicals may cause some pain, okay? Certain prostaglandins and leukotrienes could, but so chemical from the bee sting may be going to hurt because it's a toxin. So some of your pain comes from that. But in the inflammation, the inflammatory responses, it's twofold. It's like chemical, and the neurons in that area are squeezed by the fluid and the pressure. Imagine if you have enough pressure to raise your skin up, that's a lot of squeezing going on in there. You have neurons in there. It irritates those neurons. And so the pressure tends to cause the pain too. So that's redness, swelling, heat, and pain. Now I'll go back to that former slide, however far it is. They have started to recently say that there is sometimes considered a fifth sign. And the fifth sign would be the loss of function. Loss of function. Okay, let me ask you this. If a bee stung you on your elbow, would you really want to move that elbow joint a lot? Why not? Because it would hurt, right? Because you have inflammation in there. So the pain actually has a purpose in this case. If you have damaged tissue, you need to heal that tissue. if you move that tissue, it won't heal as well. So the body, in part of inflammation, causes pain to keep you from damaging the area further, to keep you from losing it so that it can heal, which to me is pretty smart. We hate pain, you know, but sometimes when you have damage to the body, if you use the body, you will hurt yourself even worse. So the body is trying to protect you and say, stop that, quit doing that, it hurts. Take a break. But, you know, we take Tylenol or steroid shots and go do it again and again and again, get more inflammation, and just keep the cycle going on and on and on. And that's a whole big table that basically says what I just told you guys all about. This is kind of a cool picture right here. We are still on which type of immunity? Non-specific. Look, that's not a bee stinger. It's a nail, tack, whatever. It could have been a splinter. It could have been a staple. It doesn't matter. When it breaks through the barrier, the body is going to try to do certain things to it. That's just the way that it works. As a part of this, we need to understand, which you guys already know most of this, would it not make sense for white blood cells to move into the area? Right? If you have damage, of course that makes sense. So here's kind of the process. Notice that when you have damage to this barrier, you get what's called a leukocytosis. A little bit. What does that mean? You know the term, I think. Maybe not. Thought I gave it to you. Just an increase in white blood cells. That's all. You get more white blood cells. Why would you want more white blood cells? Because you're in danger of things getting into your body through the broken barrier. Now check this out. The white blood cells, and this is amazing. You should Google this, and you will see, maybe that has some videos showing it. The white blood cell actually starts to stick to the wall of the capillary. As it's rolling through the little capillary, it starts to stick to the wall. It slows down. That's called margination, when it sticks to it and slows down. The wall of the capillary, the internal wall of the capillary. Because it can't get out if it's moving real fast through the blood, correct? So what happens is it starts to stick to the capillary wall, and then it will squeeze down and crawl out. And that's called diapedesis, which we did mention before. And then, just like all these concepts you learn in A&P, you use them over and over again for all kinds of stuff. Hey, if this is where the infectious material is that was introduced on this nail or tack or whatever it is, they secrete chemicals, right? And these chemicals, wouldn't they be in a higher concentration here? You know what I'm saying? Where they're produced, they're higher. And then they would spread out and be in a lower concentration in other locations. So here's what happens. The white blood cell gets out. It touches the chemical. It starts to contact the chemicals. It gets a chemical scent. And it will start to move where? Towards the higher concentration of that chemical, whatever it is. That act of moving to where the inflammation is called chemotaxis. The following of a chemical trail. Chemotaxis. Just like a bloodhound works, right? You give a bloodhound a shirt of somebody and you tell them to go chase them, and what are they going to do? They're going to follow those chemicals, And then they're really going to start to bark when they get closer. Why? Because the closer they get, the less dispersed the chemicals are, the concentration gets higher, and they can detect that. So your white blood cells are kind of like little bloodhounds going after the bad guys. That's pretty amazing. And very detailed. So what we've done is we've gone from the barriers to the phagocytes and some chemicals. And now it's time to talk about some more chemicals. The next chemical is a specific kind of chemical. Sorry, I should never use that word now, because we're doing what kind of immunity? Non-specific. This particular chemical has the name interferon. Interferon is classified as an antimicrobial protein. Just think in your brain, what does antimicrobial mean? Against a microbe. Is a microbe a very particular little term or not? Is it kind of generalized? It's generalized against any foreign cells that would get into your body. We could call them microbes. We could call bacteria microbes. We could call parasites microbes. We could call viruses microbes. So what we're saying is this chemical interferon is a protein that is produced against these. So let's talk about how interferon works. And I think this picture is fabulous for talking about it. One of the main things that you want to understand about interferon is that it is protecting your uninfected cells from a virus. It is protecting cells that have not yet been infected. Everybody understand that? When we did natural killers, it was destroying your body cells that were already infected. It was causing your cells that are infected to get destroyed. Okay? Those cells are infected, they're going to be doing bad things, so we just need to get rid of them. That's what natural killers do. So let's think of this process. If you have a virus, and this is just a little bit about how a virus works before you get to micro, okay? Because many of us don't really know how a virus works. The basic concept of how a virus works is a virus gets into your cells, it kind of takes your cell hostage, and viruses cannot reproduce. They have to use your cell to reproduce. So look what they do. They go in and it messes with, you can see this, it messes with your DNA, which is what's crazy, and it inserts itself in there, and as we know, DNA is used as a blueprint to build proteins, right? Well, those proteins are important in the virus. So the virus is trying to get in there, take our cell hostage, use our DNA and machinery to build new virus. Do you think your cell wants that to happen? No. It doesn't really want that to happen. So here's how cool our cells are. When that happens, when a virus takes them hostage, they recognize this. Kind of sad. That cell has been infected. It knows it's been infected. Its best thing it can do is try to save its neighbors at this point. It can't save itself, so it wants to save its neighbors. How does it save its neighbors? By secreting interferon. Yeah. So interferon is produced by virally infected cells. Interferon is produced by our own body cells that have been infected by a virus. Does the interferon in any way protect that cell that has already been infected? No, it does not. What it does is it tells the cells in the neighboring region what the bad guy is, and it allows them to block the viral replication. Okay? So it's pretty cool. I mean, is that going on in your body? Yes, that is kind of going on in the body, but it's not exactly that. That would be one thing, but that's what's called specific immunity, which is a little bit different. Okay? So, big idea, interferon. Produced by what kind of cells? Infected cells. That means your cells, right? Your cells that have been taken hostage are infected by a virus. Do they protect that infected cell? No, they protect which cells? The neighbors, the uninfected ones, so they can stay uninfected. And how do they protect them? By blocking the ability of the virus to replicate or reproduce itself inside of them. That's the big concept. There are many different interferons. Interferons are used as drugs now against all kinds of stuff, but that's the basic concept of how they work. Okay, next chemical. You can turn your page to see this chemical. I like the picture that looks like that. While I talk about it, I'm going to go through the different slides with you, but in your book you might want to find this picture, okay, that looks something like that to look at while we go through this. Complement is a group of about 20 inactive blood proteins. So I like to build a big picture as we go through this and remind you guys of a certain thing. And that would be the fact that proteins are how important to your body? They're amazingly important to your body. Collagen's a protein. It's 1/3 of the proteins in your body are just that one protein, collagen. Some hormones are protein, right? Certain neurotransmitters are protein-based neurotransmitters. Now we're learning that these chemicals, like interferon and complement, immune system chemicals, are proteins as well. We know that proteins are channels into and out of cells. We know that enzymes are proteins. So as we start talking about the body, we're building our knowledge of, wow, proteins are the real workhorses of the body. The functional proteins do tons of stuff, and the structural proteins give us our foundation that we build everything around. So proteins are totally important to our life. Well, here's another one. We have proteins, and we know we have plasma proteins that circulate through the blood all the time, right? This is one of those. Complement, in the inactive form, circulates through the blood. Okay? Now, there's lots of names for the different complement proteins. I don't get picky on the names. You can see how they're named. Complement C1 through C9. No big deal. I'm not going to ask you about that. That's not what kind of our purpose is right here. But here's kind of what complement does. Its job is to amplify inflammation. What does amplify mean? Yeah, to make it stronger, to increase it. It's going to support inflammation. It's going to stimulate more inflammation to take place. It's going to stimulate cell lysis. And what was cell lysis? Okay, it's when a hole is poked in the membrane. The cell swells up because the fluid rushes in. Why does fluid rush in? Why doesn't it rush out? Because solute sucks. Because that cell must be full of solutes, right? And there's enough solutes that it's going to pull the fluid in by diffusion, like we know, and it's going to swell up and it's going to explode. So that's cell lysis. We'll talk about how it happens. It's different than the one that we just mentioned a while ago. Okay. And it tags whatever it happens to be attaching to this complement. It marks that for phagocytosis. It marks an object, a microorganism, what have you, for phagocytosis. So here's the memory device I want you to write down. It's a few letters. Write O, and then a hyphen or dash, however you want to say that, and PICL. O-P-I-C-L. O pickle. I know it's silly. Just go with me. It'll help you remember it on test day. What do you think the P is? Complement stimulates phagocytosis. What's the I? It amplifies what process? Inflammation. And what's the CL? Cell lysis. So if you remember when you're talking about complement, remember O pickle. I'll tell you what the O is in just a second, but you'll have it stimulates phagocytosis, inflammation, and cell lysis. Right there. Now, what I haven't told you yet is that there's a special name, and I'm just going to put one word up here, that I kind of skipped over earlier because I wasn't ready to just give you vocabulary because I don't like doing it like that. Anything that tags or marks something for phagocytosis is known as an opsonin. An opsonin is any chemical or object that tags or marks something for phagocytosis. So would you consider complement, which is a protein that marks something for phagocytosis, an opsonin? Yes. So the O and then pickle tells you that this is an opsonin because it stimulates phagocytosis. It also happens to enhance or amplify inflammation and causes cell lysis. Now the cell lysis part of it looks the same, but the name is different. So look at the picture. Here's the picture. As complement is activated, those complement proteins inject themselves into a cell membrane like you see right there. But what they form this time, instead of the chemical that we already talked about, this time that entire complex is known as a MAC, a MAC, or membrane attack complex. All right. Well, if complement's inactive, that means that this isn't always happening, right? It has to be activated. So there's two pathways right here. There are two different ways that complement can be activated. And since we're doing non-specific, I'm going to focus on the second one first. The alternate pathway. When complement proteins, and I'm going to make it simple, when there's the presence of a microorganism and complement proteins interact with that, it stimulates the activation of the complement cascade, which means one complement becomes activated, it activates another, and another, and another, and another. So I'll say it again. Just simply the presence of a microorganism, so bacteria, that happens to be in there, that complement binds with or attaches to, triggers this response. So what am I really saying? Basically, the presence of microorganisms that happen to come in contact with complement causes this. That's cool. That's pretty basic and pretty easy. Notice it says microorganisms. Notice it's non-specific. That means it doesn't really care which one it is. It doesn't have to identify the exact one. It's like, okay, you're a bad guy. You're inside here. I'm going to tell my buddies, other complement proteins, I'm going to activate them, and here's what they're going to do. Some of them are going to try to poke a hole in you so you explode. Others are going to bring an inflammatory response to try to wall you off and separate you from the rest of the body. And still other ones might tag you so that other white blood cells can come in here and eat you up. That's the three things that would take place from that. Okay, the classical pathway, all I want you to understand about that right now is that is part of specific immunity, which it does no it has no purpose for me to run through that with you now because we don't even know how specific immunity works. So in your notes, complement is activated two ways. By the alternate pathway, which is nonspecific, which is just simply the presence of a microorganism, and by the classical pathway that is specific that we will have to talk about later. Membrane attack complex. Membrane attack complex. It's right here. It's those proteins. You see this right here? This is one of the MAC proteins. It is formed when the complement starts to activate that cascade. It forms these little proteins that inject themselves there. So the whole thing itself then is known as a membrane attack complex because it's a complex of proteins that has poked a hole in the cell membrane. It is very much similar to perforin. That's what we said perforins do, right? But they just gave it a different name with complement. But it does the same thing. It does the same thing. All right, the last part of nonspecific immunity, then we will take a break and come back and do specific immunity. The last part is fever. Raise your hand if you've never had a fever. I would worry if you never had a fever. Thank you, Andy, for making me worry. Since you've all had one, tell me what it is. It is an elevated body temperature. Why on earth would your body want to do this to you? Because as we increase the temperature, what does that do to certain proteins? It alters them, and the name of altering a protein is denatures. Do you remember that from AMP1? It denatures proteins. And the concept is this. If we raise the body temperature enough, we denature the foreign proteins that are in our body, which would be maybe the proteins in the bacterial wall or the viral wall, or maybe even some of the chemicals, the harmful chemicals that have been released. So it would tend to neutralize those and make them ineffective. It's brilliant, really. The problem is, if you heat up too much, you start killing the good ones too. But how much is too much? Yeah, 105, 106, right? At 105, 106, your body gets hot enough to actually start to denature the proteins in your brain. Does that sound yummy? That doesn't sound yummy. What is a good visual for protein denaturing? The best visual there is that I can think of. Good try. That's not how I think of it. Here's something to think of. Remember, it's just altering them chemically, right? I know we like to think of, when we're thinking of destroying and white blood cells killing and stuff, we think of melting and oozing and stuff like that and exploding, right? But let's take it backwards and let's think of this. Lots of proteins are gels or semi-solid or in a liquid environment, right? They're not all little chunky little pieces of steak. So visualize an egg white. And visualize what happens to the egg white when you heat it up. Does it change its state? Yeah, it goes from a gel to a solid. Once it does that, can it ever go back? No. So whatever function it had in that gel state, let's think about bacteria now, you would totally permanently alter it with the temperature change like this, and it could never go back. So you would render it useless for that purpose. That's cool. Okay? Just like the egg, you can't throw it in a blender, add water, and turn it back into the gel-like egg, can you? I mean, you can beat it up all you want, but it's not going to go back. It's already been altered. That's how fever works. It does a couple other things, too. What you need to understand is this. White blood cells secrete chemicals called pyrogens that resets your body temperature your body's thermostat, hypothalamus, however we want to say that, and raises the body temperature. Those chemicals are called pyrogens. Any pyromaniacs in here? I hope not. What does pyro mean? Fire. What does gen mean? Create, produce. Gen means producer. So a pyrogen just means a fire producer. What's a fever? It's a little baby fire inside of you, heating up. Now this is pretty cool. It says it denatures it, but look, here's the benefit of a moderate fever. In addition to what we've already said, this is so cool, it increases your metabolic rate, which helps to speed up repair in your body. Increases, yes ma'am. Yeah, let me get this little part and we'll go there, okay? We'll totally go there. The other thing that it does is it causes the liver and your spleen. Two kind of neat organs, right? Liver and spleen. Liver has, yeah, it's amazing. Liver has like 500 different functions, and they knew 500 functions of the liver 20 years ago, so how many do you think they know now? Man, I don't even want to know how many they know now. But anyway, the liver and the spleen hoard iron and zinc, which are what certain microorganisms use to divide and multiply. So your body is so smart that when the fever happens and the temperature goes up, the other thing that these pyrogens signal is they signal iron and zinc to be taken out of your system, basically, and start getting stored in organs so it hides from the bacteria that want to use it to multiply. So you're basically shutting down part of their process that allows them to replicate. I know. See, I have a problem with that because I don't understand that. And I am torn by that. I am torn. And no, I'm not repeating that for you guys. Now you can think about it. She said, why do you take zinc then when you're sick? I don't know, because your body's going to try to hoard it when it goes in there. You know, I mean, I know that there are benefits from zinc, but if you're really going to want to hide it, since I learned about this, I don't take zinc anymore when I get sick. That's the whole point for me, is what it boiled down to. Okay, your point. Ask me again. All right. So when we have fever, some people like to stop the fever right away, you know. And as long as you remember that fever is there for a purpose, you obviously have something going on in the body. So the time that you really want to fight this, there's a couple of ways to look at it. If you have a young kid and your kid's been 102.4 all day long, maybe they need a little break from that. You know, for 8 hours they've been 102. Maybe they need a break. 102 is not enough to denature your proteins. Okay, it's pretty low. But for a super long period of time, it's not good. But if they've been 99, 100, 101 in that range, it's kind of one of those judgment calls, because that's the body showing you that it's fighting. And we're not just talking kids, we're talking ourselves too. Okay? Different people's bodies are different though, because they're different people. And we have different genetics. And some of you guys get sick and you immediately get a high temperature. Who does that? Anybody in here? When you get a fever, you get a hot fever. Other people will get a low-grade fever, a 99.9 or 100.1, and you'll sit at that forever. Point is, maybe take something if you feel like you need a break from it, but don't jump on it too fast, because you'll stop your body's mechanism for trying to heal itself. It's kind of a fine line. So it's a judgment call on how the person feels. It's a judgment call on how healthy the person is as an individual. If they have other disease conditions or something, you may want to help them out and give them a break from it too. But it's not so cut and dry, and it's definitely, we really can say, if a kid has had a 100-degree fever for 2 hours, they don't necessarily need a break from a 100-degree fever, because that's their body fighting it. All right. Unless, of course, their normal body temperature is 97.1. Like my body temperature is normally 97.3 or 4. So if I'm 100.1, guess what? That's like somebody else being 101.8 or something like that. So you got to know your set point to know what's actually a fever for you. If I'm 99.2 or 3, I got a fever. It's like an average person being 101, basically. All right. That is non-specific immunity. Let's take a little break. Let's load this little talk up.